Hello, everyone, and welcome to the Interfex IPS Virtual Technical Tour. My name is Tim Schuster. I'm the Director of Process Engineering at IPS, that's Integrated Project Services, and I will be your virtual tour moderator today. The virtual tour will have three suppliers from the bio field for this presentation. Presenting today will be Pall, YMC, MilliporeSigma. I will introduce Ian Sellick from Pall, Gerard Gach from YMC, and Joshua Hayes from Millipore, who will do presentations of their products and services. We'll share all this important links at the end of the virtual tour for the attendees to access any additional product or company information. Let us begin by welcoming and introducing our first presenter today, Ian Sellick. Ian is the Director of Marketing at Pall Biotech. Ian will discuss some new product offerings that increase the coverage of Pall's integrated solutions and platforms for end-to-end processing of proteins. Welcome, Ian. Please begin.
Thanks, Tim, for that introduction. As Tim said, I'm Ian Sellick from Pall Biotech, and I'm going to go through a little tour in maybe a somewhat of a different way as if we were doing this in person. Historically, we have always done a journey through our booth, and we're going to stick to that kind of theme. But what we will do during this very short presentation is to introduce you to some of the new products that we either have launched recently or we're planning to launch at Interfex. And we're going to look at some other aspects of our business and how that helps and how we interact with you to accelerate your process and your time to market. So this is part of our Your Journey, Your Way theme, because we have a number of different ways in which we can assist in end-to-end processing, whether your requirement is for an all single-use solution, a stainless steel solution, some hybrid of both. And just as a preview at the bottom of the screen, you can see some of our latest products which address an area that we have typically got as far as processing your bulk material, getting it ready for sterile filling, but not necessarily had the option of bulk packaging, that shipping, freezing it, and those sorts of activities, which are so necessary, particularly at the moment, where very large quantities of vaccines and pharmaceuticals and therapeutic products are being made and need to be sent off to filling companies that may be in a different country or even in a different continent. To start with, let me take you back to last year, and so what you're missing, of course, is the ability to actually see and touch all that nice, shiny, new equipment. The most important part of the booth at all times are the people, because without the people there, you really can't get a proper understanding of the equipment and how it integrates with your process. And the booth historically has been a place where we can interact with you and ask questions and ensure that we actually are guiding you in the right direction. And that has not stopped in the virtual world. All of our people are available on a global basis, and in fact, we've been adding people quite significantly to make sure we have full coverage across the world for every opportunity and every project that you're working on. And historically, we've been able to do that, as you see on the other screen on the right. We've been doing that virtually for some time, and we've accelerated our program of virtual design and product application offerings. So, with the people, all of whom are very highly trained, very thoroughly versed in all of the equipment and the products and the processes that you're trying to bring to market. We have whole teams everywhere that you need them, and this basically leads into our service offering, which we feature at all of our exhibitions and events, and really brought this to the fore in our ability to offer you process development services through global centers of excellence, and building a process for you that shares our expertise with your needs and turns over a transferable process. And we've done this in the current situation a number of times already, most of which are in the public domain. And if you look online for our response to COVID-19, you'll find quite a lot of examples. Now we have a very large portfolio of ready configured equipment. Plus, of course, we do a lot of custom work, and we can help you pick and choose and actually develop processes with the whole suite of equipment that we have available, all the way from the bioreactor through to final filling and finishing. And obviously, don't have time to go through all of the pieces of equipment which are available to you, but just gives you some idea of the range. And one of the areas that we have recently developed is the ability to do bulk filling into containers that can be used for simply holding in your facility, or for freezing and shipping anywhere that you need to send that material around the world. So if you have any questions about any of this, any part of the process, or you want to discuss an entire end-to-end process, whether you are starting from cell culture with adherent cells or suspension cell culture, we have the solutions and the capability to help you. And a large number of fairly standard bioprocessing platforms that you can draw upon, whether you have a recombinant protein or a monoclonal antibody-based process, you're working with viral vectors in which we have a particular expertise, or some other virus, adenovirus, or maybe you're in the cell therapy part of the marketplace and you're working with things like oncolytic viruses, or some of the newer cell therapy technologies based on mesenchymal stem cells, for example. So we offer all of this at scale, at the sort of volumes that are being demanded by the market these days, and a huge amount of expertise in the teams of people that we have available for you. What we're offering is a single integrated solutions provider approach that helps you to scale your process and reduces manufacturing risk, and turn over to you a very tech transfer package provided by teams of professionals who have done this many times before. So obviously, manufacturing readiness is critical to speed to market ambitions. We're seeing that particularly at the moment, and we're working with a very large number of you who are hopefully listening to this broadcast to help you get critical products to market at the moment. And using the platform approach really is a key to supporting the speed to market, and having a single integrated solutions vendor does offer truly strategic benefits in having a fully integrated set of equipment. Thank you very much for listening to this, and feel free to contact me or Pall Biotech any needs that you may have, especially during the current times. Thank you.
Thanks, Ian, and thanks for sharing the technical resources available at Pall Biotech. Our next presenter is Gerard Gach. He's the Chief Marketing Officer at YMC Process Technologies. Gerard will discuss YMC's continuous chromatography and how continuous chromatography can benefit end users. Welcome, Gerard, and please begin.
Thank you for the nice introduction, Tim, and thank you to IPS and Interfex on behalf of Burkert and YMC for arranging this get-together. You've been really great to work with, and thanks to our audience for stopping by on this virtual event. In the next few minutes, I'll give you some insights into a next-generation downstream technology that has quickly expanded into GMP production, and some insights about how our technology supply partner Burkert helped us to achieve one of the more critical to quality attributes that has made the scale-up of our Continuum Twin Continuous Low Pressure Liquid Chromatography Platform, or LPLC, so successful. As you may know, the transition from batch processing to continuous processing is understood to significantly lower costs of production, increase quality, and availability of drugs. Continuous manufacturing has been proactively promoted by the FDA over the past few years. Indeed, upstream processes have made great strides towards continuous production with high titer, fed batch, and perfusion-based cell cultures. The higher titers fed to the downstream purification area have thus caused a bottleneck in the downstream operations, as they were designed primarily for batch processing of lower concentration feed streams. This bottleneck has been especially seen in the first major downstream purification step of capture chromatography. To overcome this productivity pinch point, YMC has leveraged a patented process called Capture SMB, which was first developed by a Swiss company called Chromacon. Capture SMB, long proven in the lab, allows continuous purification of mAbs, recombinant proteins, and other biomolecule therapies using two columns chromatography versus the conventional single column chromatography approach. An example of such a smaller scale continuous chromatography system is pictured here on the left. Let me first talk about what this system accomplishes, and then I'll illustrate how it works. Employing this, or frankly, most any multi-column chromatography system brings the user an increase in productivity of 2 to 3-fold, savings of up to 50% in protein A resin, which, by the way, can amount to millions of dollars per year, and decreased buffer consumption up to 60%. Again, if you take a look at the buffer consumption, that savings can be measured in the hundreds of thousands, if not 7-figure cost reduction. So how does it work? So I'm going to distill a 30-minute control automation process configuration explanation into 30 seconds. So hold on. In conventional single column batch, product is loaded onto the chromatography column until just before the target molecule or the valuable drug product breaks through. The product protein loading pattern is shown here in the solid yellow. Not wanting to lose any product, the operator necessarily stops the loading of the column before product breakthrough and loss. This results in only 40 to 60% of the protein A resin, which is typically used in the pack column, being used. At 13,000 or more per liter, this is a significant underutilization of this expensive resource. What YMC Continuum Twin LPLC process with Capture SMB does is it splits the column into 2 separate columns of shorter bed height, usually around 10 centimeters, and allows each column then to be used to nearly 100%. And at the same time, the user uses much less protein A resin or can process at a much faster throughput rate. As you might imagine, the management of the fluid streams in a 2-column continuous system where the process is switching frequently between 2 columns versus a single column batch system requires much better control, as well as a very tight tolerance of holdup volumes. So valves were a component that we identified as critical to the successful management of our twin column chromatography process. We had had much success with valves, block valves by Burkert in our standard batch chromatography systems. So we knew that they were generally up to the task for many of the twin column chromatography duties. But in certain flow paths for this new technology, flow paths that were critical to doubling or tripling productivity, we needed not only reliability for the continuous operation and multi-directional flow management, we needed an extra level of precision and reduction in holdup. Here at the top, you can see that a conventional valve arrangement has about twice the dead volume as a block valve, shown here in the middle diagram. But what we required was beyond even the performance of the block valve. So Burkert introduced us to a design they call Robolux, illustrated here on the lower part of the slide. That has even a smaller body and has a dead volume that is time again smaller than the typical block valve, which is shown in the middle of the screen. So this Robolux design gave us the ability to achieve the rapid switching that we desired, but with far less holdup, helping to shorten the cycle time while preserving the highest gains that were possible by the capture SMB enabled twin column process. So the result was able to take this bench scale process and with users such as Bristol Myers Squibb, prove the ability to scale the technology to GMP production, where now these units have been successfully producing clinical material. At the heart of our patented two-column approach is it's much more simple than other multi-column designs using 3, 4, or more columns. But we also built into the unit, and this is important, additional functionality, essentially combining 4 unit operations into a single skid that can do batch chromatography, it can do continuous chromatography, it can do sequential orthogonal polishing chromatography, which is poolless, with the same skid. And additionally, it has buffer inline dilution capability, allowing the dilution of buffer concentrates, which further reduces the operating footprint. End users have published extensively on productivity gains and have characterized the performance with the outcome of now multiple of these units operating at flows up to 20 liters a minute on GMP proteins. One last feature that also leveraged Burkert technology, along with a unique on-off disposable single-use to stainless steel connection by Millipore, is a proprietary inlet and outlet design by YMC that allows this unit to be operated in a single-use suite as a completely closed and sanitary system. Thus, you gain the advantage of the precision that only a durable or a stainless steel system can deliver, but in a lower classification, lower cost, single-use operating suite. Okay, I've covered a lot of ground in a short time. If you want further insights on any aspects of this technology or our other standard and custom downstream skids, I'd be happy to follow up with you, or you can grab a bowl of popcorn and an adult beverage and sit back and tune into the YMC Process Technologies YouTube channel, where we have a lot of video seminars on a multitude of topics like this. Or you can just ping me at info@ymcpt.com. So special thanks to IPS and Interfex for arranging this get together. A lot of work went into organizing, but I think IPS and the Interfex crew are doing a great service to our industry in this unique time. I'll leave it there for now and turn it back over to Tim.
Thanks, Gerard. And thanks for telling us how continuous chromatography can increase productivity and decrease cost. Our final presenter is Joshua Hayes. Joshua is the Associate Director of Commercial Strategy, the BioContinuum Platform for Next Generation Bioprocessing at MilliporeSigma. Joshua will discuss products and solutions that make up the MilliporeSigma BioContinuum Platform. Welcome, Josh, and please begin.
Thank you, Tim, for that introduction. Hi, everybody. My name is Josh Hayes. I'm part of the BioContinuum Platform team here at MilliporeSigma. Thanks for taking a few minutes of your day to join us to talk about what's new with us at our virtual booth. I do wish I could be seeing you in person. I do enjoy that part of the annual tradition every year in New York City. So hope you're all well, and thanks for taking some time to talk about what's new with us. As I mentioned, for MilliporeSigma, the BioContinuum Platform is really our newest innovation, and it's a brand name that we use for all of our forward-looking future manufacturing type technologies. And it really consists of 3 parts: upstream, downstream, and digital. And we'll spend a little bit of time talking about each of those components over the next few minutes. But I think it's important to understand our fundamental philosophy within the BioContinuum Platform, and that's really in response to where our industry is headed, creating technologies and best practices that help us get there, largely driven by the tech notes that were released by DPOG a few years back, the tech roadmaps. And really, most of us are used to thinking of our industry operating in discrete unit operations. And the trend for many is shifting towards more connected and continuous manufacturing, combined with things like inline analytics that are now moving testing from offline to at-line and ultimately inline, and leveraging single-use technology. So migrating from stainless into single-use is not a new concept. People have been doing it for years. But when we combine best-in-class process technologies to intensify processes and connect them with inline analytics that are leveraged and really made possible through automation and software advantages and advances with single-use technology, it really springboards us as an industry towards being able to build this facility of the future. So again, upstream, downstream, and digital for us are all areas that we're focusing on bringing some innovation to market. And I would like to just talk briefly about each of those today to let you know what's new. On the upstream side, we have a few exciting things that we have recently launched. High cell density cryopreservation in order to help customers bring high density process intermediates from location to location to really increase their efficiencies on the upstream side within perfusion. We have a specific perfusion media called Solvento 4CHO X that's recently launched. We have made alterations to our single-use bioreactor portfolio to accommodate perfusion, which is where many people are headed as they move down the path towards continuous manufacturing. And last but not least, we are introducing some cell retention technology to the market under the name of Sellick. So this one in particular, I wanted to spend a few more minutes on, just to let you know that the small benchtop version has recently launched and larger versions will continue to launch later this year and into early next year. But it really is a nice solution on the cell retention front to give our customers in the market another option with great performance. We've seen some fantastic results with less than 0.02% cell passage. And we've had greater than 95% cell retention, which is of course very important with a cell retention device. It's very easy to set up, and I think also equally importantly is completely linearly scalable, which is important in the upstream space as well. What you do at the bench needs to translate to the 50, 200, 500,000, 2,000 liter scale, etc. On the downstream front, we have recently introduced our buffer delivery platform. I think that we're looking at it as a platform which ties in with our convergence theme of looking more holistically at manufacturing processes. And as a platform, it's intended to encompass all the things that you would need to perform this step. Many folks in industry have entire buffer preparation suites and teams, and we recently introduced this platform, which is equal parts system and chemicals, if you will. So it's the system itself, it's the buffer concentrates, which we make at various sites around the world. It's the single-use assemblies that allow transportation of those concentrates to the site and storage once they're diluted, as well as the associated services and training to use the system. And from a technical perspective, we really have opted to focus on volumetric flow control because we believe it provides some advantages over pH and conductivity, for instance. Those are historically what a lot of people use to measure their dilution. But we've gone with volumetric flow control based on the Lewa Ichthys pump, which is very, very accurate, less than 1% error in the accuracy for the flow control. We have done a wide range of buffer concentrates from within our chemical portfolio. We do find that sweet spot to be about the 20-to-1 dilution ratio, but it really can provide some impressive economic benefits to consider on-demand buffer preparation like this. For instance, we recently designed and are building a new manufacturing suite in Shanghai, and we estimated that almost 20% of the floor space could be saved by shifting to on-demand buffer preparation versus a dedicated buffer farm, for instance. Shifting gears briefly to the digital side of things, this is where we're really bringing some new and exciting innovations into the market. First, I want to mention a little bit of a preview for a smart control platform that we're bringing to market later this year. And the intent really is to shift from the classic, very manual way that data is collected into a much more automated fashion. So on the left, you'll notice there's a person in the middle with a manual batch record, pulling information, writing data points down, capturing settings and things like that from the systems, from tanks, from batch records, or from raw materials, lot numbers, things like that. And really, we want to move towards automating that so that there's this continuous flow of information into and out of this platform so that everything is centrally located and the data can be pulled and analyzed much more quickly and efficiently, which allows people to make data-driven decisions, which are a much better way to optimize their manufacturing efficiencies. And I think most of you are probably familiar with the fact that today, much of what we do is still on paper. And it's not going away anytime soon, but we're moving in that direction, collecting data, pulling information from various sources in a manufacturing environment within the quality labs, scale-up labs, PD labs, et cetera. A lot of different sources of data being collected. Equipment doesn't always talk to each other. Everything is reviewed on paper oftentimes with very manual process control. But in the future, we want everything to be centralized. Having it centrally located and easily accessible offers a tremendous number of advantages, as you can imagine. Really, it speeds up scale up, it speeds up tech transfer for those that are manufacturing with CMOs, et cetera. And really, as we make advances in the software and automation front and associated analytics, we can really start moving forward much quicker and start talking about things like predictive maintenance, just-in-time training, things of that nature that we've been talking about for years, but are now really becoming possible with the advances in technology. And lastly, I'd like to spend a couple minutes talking about our newest offering in the software space, ProcessPad. This launched at the end of April in 2020 under our new brand name, the Bio4C Suite, which is where all of our digital technologies will reside. And ProcessPad really is unique in that it is a browser-based data aggregation and visualization engine that will really help facilitate our industry moving forward and really make use of all of the data that's generated in a manufacturing run. And today, I would argue, is poorly stored, very difficult to access and make cohesive. And I think really that what I would highlight with ProcessPad, and I'll turn on my laser pointer to help showcase it, is it really is intended to sit at the center of most of the data that is being generated in a manufacturing environment. So again, there's manual records that can be uploaded into ProcessPad because paper's not going anywhere, QC records, PD records, things like that that are paper. It can connect to historians and therefore pull real-time machine data into the system as well. And it can tie into external databases like LIMS, QMS, et cetera, all of which sort of allows it to sit at the center and become the source of truth for data. So it pulls all these data from very disparate sources into a central location, purely web-based, that allows people to go in and access it with a few clicks of the mouse and analyze it, generate meaningful reports and visuals so that they can communicate with themselves or their stakeholders how things are going, and they can speed up investigations, continuous process verification, all these things that are part of their landscape become much more efficient with ProcessPad. And in closing, I wanted to make you aware of the campaign that summarizes a lot of this information. I would encourage you to point your smart device at the QR code on the screen, and it will launch a nice website which has some really interactive augmented reality features to help you learn more about what we're doing in the BioContinuum platform. And as always, if you have any questions, please don't hesitate to reach out to your local MilliporeSigma rep, and we'll be happy to continue the conversation. So thanks again for your time and have a great day.
Thanks, Josh, for your presentation. We want to thank all our Interfex IPS BioTour presenters, that's Ian, Gerard, and Josh, for the contributions to this virtual session and sharing their company's product information. We appreciate all that have virtually attended this tour today. To contact the suppliers you heard from today or access these presentations, please go to interfex.com. Thank you all again, and please stay safe and have a great day.